non-smokers. I had told you that. But there's also no difference in the intensity per cell in female smokers compared to smokers, whereas there is in male smokers compared to non-smokers. And so this is where we're starting to see sex or maybe gender differences. So we don't have the ability to tell how heavy of a smoker these people are. And gender differences might come into play, whereas the males who do smoke might tend to smoke more than the females. And so that's why we might be seeing a greater difference in men compared to women in terms of polysialation in these T cells. So we don't see sex differences in healthy, but we do start to see some differences in people who have this chronic inflammation. But we want to know what is happening. Why is this happening? And one of the challenges is that we have no idea what proteins are polysialated in a T cell. None have been identified so far. If you look at an immunoblot of a T cell lysate, so this is from a study that we did that we published in 2023, where we were actually developing a inhibitor of polysialation in collaboration with Steve Withers at UBC. And really, it's just the DMSO control that's important here. These are T cells several days after activation, in vitro activation. So they're isolated from PBMCs and then activated in vitro. And you can see lots of different proteins. If you treat with the endo-ide control, you lose all of this reactivity. So there's lots of stuff in here that looks like it could be polysialated. And if you look at the known polysialated proteins, it's a short list. Unlike monosialic acid, the list of known polysialated proteins is short. CD56 is that iconic one. And it is possible that some of this is CD56 because it is re-expressed in some activated CD8 positive T cells. SYNCAM is not present in T cells. Neuropilin-2 is not really present in T cells. CCR7 is a protein that was identified in dendritic cells, and CCR7 is really interesting. I'm going to come back to this protein. It's a chemokine receptor. So in dendritic cells, polysialation of CCR7 is required for the migration of dendritic cells towards the chemokine ligand CCL21. And it's a specific interaction with CCL21. CCR7 has another ligand called CCL19, and it doesn't matter if CCR7 is polysialated, it will migrate towards CCL19, no problem. The migration towards CCL21 is polysia dependent. So this is actually present in T cells, but it's present in naive and central memory T cells. It's one of the markers for naive and central memory T cells. And if you recall, naive T cells had no polysia. Central memory did, so maybe some of this is CCR7. E-selectin ligand 1, this is a possibility. ST8-SIA2 is not expressed in the immune system, and then ST8-SIA4, this is the polysialotransferase, which also autopolysilates, is. So we've got 1, 2, 3, 4 proteins that really can't explain this. And so we wanted to actually do an experiment that would allow us to identify the polysialated proteins here. So most people would think that immunoprecipitation is the way to go for identifying protein of interest. But one of the challenges with polysia is that it's really large, it's negatively charged, and lots of proteins in a lysate might bind, anything that would bind to a polyanion, ribosomal proteins, DNA binding proteins. The immunoprecipitation proteomics tends to be really messy. What I'm showing you here is data from Nick Stamatos' lab, and it was published in 2007. And this is the paper where they identified neuropilin-2 as one of the polysialated proteins. And what you can see, if you look at this data, is the quality of the data is poor. So in this paper where they identified neuropilin-2, they only saw one peptide. And they found a couple of other proteins. They actually found CCR7 in this paper, but they ignored it at the time. This was before CCR7 was identified. And they also found this protein down here, MGAT2. We subsequently found that this is also polysialylated. So they actually had data. The data is not bad, it's just not robust. And so we thought we could develop a better method to identify polysialylated proteins. And that's what we did. So we took the polysialectin. This is the hydrolase that has a double mutation that makes it a really strong binding protein, but it doesn't hydrolyze the glycan. And what's fabulous about this protein is that it comes from the tail spike of a phage that was discovered in sewage. You can do anything to this protein. You can heat it, you can put it in SDS, you can do anything to it. It is robust and it will continue to bind polysialic acid. So what we did was to engineer it so that it had an AVI tag and a his tag for purification. This AVI tag is a 15 amino acid sequence that can be biotinylated with the E. coli protein BIR-A. And so now we have a biotinylated polysialectin that we immobilize on streptavidin-coated beads. And when we mix that with a cell lysate, for example, any solution that we think has a polysialylated protein on it, we can wash with really strong detergents to remove any non-specific proteins, and then use this to do proteomics. And so we started with NK92 cells just to prove that the methodology worked. NK92 cells, this is a natural killer cell line. And we know that it makes polysialylated CD56 or polysia NCAM. And so the NK92 lysate makes both polysia and NCAM. The flow through has a little bit of polysia and this lower molecular weight NCAM, which is probably not polysialylated. So the higher molecular weight stuff is, and the lower molecular weight stuff is not. There's nothing in the wash. And then if you look at the beads, you see the higher molecular weight NCAM. If you pre-treat the sample with endo-N to hydrolyze polysia, we see no polysia. And we also see that nothing sticks to the beads. All of it's lower molecular weight and nothing sticks to the beads. So we sent these beads for proteomic analysis in collaboration with Nick Scott at the University of Melbourne. And unsurprisingly, we found, this is a volcano plot, so we're looking at proteins here, and we found NCAM is well separated from the mass. And there were 33 unique peptides, 46% sequence coverage. So that's excellent. We also found a couple of other proteins, including SD8-SIA4, that's the polysia transferase, myosin H9 and GPR15. I'm going to mention these again. I'm going to come back to these two proteins. So we knew that the method worked. The next thing we wanted to do was to look at another cell line. This is a breast cancer cell line that Karen Colley, I think it was Karen Colley, showed was polysialylated way back in 1992. But polysia was intracellular. It wasn't on the cell surface, and nobody knew what the protein was, and no one had followed up on it. So we did the same protocol and identified QSOX2 as the polysialylated protein. And again, 24 unique peptides, 42% sequence coverage. We're getting really nice enrichment of these proteins. We validated that QSOX2 was polysialylated. This is a straight Western blot where you can see a bit of a smear here that sort of collapses down here. But the QSOX2 antibody has this background here. So we did reciprocal co-immunoprecipitation. In here, we've immunoprecipitated by polysia and then looked for QSOX2. And we can see high molecular weight QSOX2 here. And then when we treat with endo-N, that band decreases in molecular weight. If you pre-treat the sample with the polysia hydrolase, you don't pull any QSOX2 down. You don't pull any polysia down. And then looking at a QSOX2 immunoprecipitation and now looking for polysia, you can see this material here, and QSOX2 is not pulled down in when the samples are pre-treated with endo-N. We looked to see if it was N or O-linked glycans, and we see decreases in signal intensity for both removal of N-glycans and inhibition of the formation of O-linked glycans, and synergy when you combine both of them. So we think that this protein has polysia on both N and O-linked glycans. Okay, so now that we validated the method in cell lines, we want to come back to the T cells. So we took T cells, we expanded them for 7 days in vitro, and then we did proteomics. And what we're looking at are 2 separate individuals here. So we see NCAM here, CD56, as expected, because it is re-expressed on some of those CD8 positive T cells. We see ST8-SIA4, which is also really exciting. One of the nice things about this method is that ST8-SIA4 reproducibly shows up in most of our experiments. That's not true for immunoprecipitation. So we're really happy to see that the enzyme that we know is there and autopolysialates actually shows up in our experiments. Myosin H9 shows up in one of these patients. This is one of those, or one of these healthy controls, sorry, not patients. This shows up in the NK92 cell line. This is a really weird protein that nobody knows anything about and has shown up a couple of times in a couple of different experiments. And if there's anyone out there who knows anything about it, I'd love to follow up on what this is. But what I'm going to follow up on is actually GPR15, because this is the only other protein that appeared in both in the proteomics experiments from both donors. And GPR15 is a fascinating protein. It's another G protein coupled receptor that turns out is a chemokine receptor. So just like CCR7, GPR15 does a similar thing. So 7 transmembrane pass protein. When it binds to ligand, there's an intracellular signaling cascade that causes cell migration. And there are a number of serines and threonines on the extracellular face of GPR15 that could conceivably be polysialated. GPR15 has no predicted N-link glycans. So we're thinking O-link glycans, which there's a ton of precedence for O-link glycans. This is not unusual. GPR15 is not normally present at high levels in a healthy adult. It is very strongly associated with smoking. It's very strongly associated with autoimmune diseases and with pathogenic inflammatory T cell responses. Its ligand is GPR15L, which was actually only discovered maybe 5 or 6 years, probably 7 years ago by now. It's a small chemokine that is secreted by cells in the skin, primarily in the skin and mucosa, although a little bit in the lymph nodes as well. And those of you who are used to looking at structures will notice how profoundly basic this protein is. Anything in blue is basic. So 30% basic residues. In what looks kind of like a helical pattern, you could conceivably imagine this polyanion wrapping around this ligand. And so we thought perhaps the GPR15L is actually binding to the polysia on GPR15, not necessarily to the extracellular face of the protein itself. So maybe polysia is a landing pad for GPR15L. We wanted to validate first that GPR15L is polysialated. This is flow cytometry of expanded T cells. What we're looking at is GPR15 here. About 50% of expanded T cells express GPR15 in our hands, and 100% of them express polysia. This is a polysia ELISA that we developed in the lab. So we use a lectin, polysia lectin. We immobilize that on a plate, and then we can pull out polysialated proteins from complex mixtures. This is a really challenging protein to work with. It's extremely hydrophobic. G protein coupled receptors are notoriously difficult to work with, but we're able to pull it out of solution and then come in with an antibody to GPR15, and then a secondary antibody that gives us a blue color. So this is a cell line that expresses GPR15. We see signal that disappears when you treat with endo-N. These are the day 7 expanded T cells. Again, we see signal that disappears when you treat with endo-N. And then this is a Jurkat cells. They are a T cell, cancer T cell line. They're not expected to express GPR15. And so we were using this as a negative control. And there's no statistical difference between these two. We can also do the reciprocal ELISA. So we put the anti-GPR15 antibody down on the plate, pull that out of the T cell lysate, and then use the antibody to polysia to look for any polysialylated proteins. And again, we see signal in BDCM and the day 7 T cells, which is abrogated when you treat with the endosialidase to remove the polysiahydrolase. So we're sure that this protein is polysialylated. And then the next obvious question is, well, does polysia actually affect the migration of T cells towards GPR15L? We have this transwell migration assay set up. So the way this works, there's an insert in this well that has a semi-permeable membrane that T cells can pass through at the bottom. So you put your T cells in the top and you put your chemoattractant in the bottom. You wait a couple of hours and then you measure how many cells have migrated to the bottom. If we put heat inactivated FBS in the bottom, we see lots of migration, and it is independent of polysia. If you remove polysia, you still see migration, which is what we were expecting. There are lots of chemoattractants in heat inactivated FBS. We were super excited to see that when you use GPR15L as the chemoattractant, we also see really nice migration. And if you pre-treat the cells with the polysiahydrolase, you lose that migration. So it looks like the migration is polysia dependent. We followed up on that by inhibiting the interaction between GPR15L and polysia potentially. So this is the exact same assay where we have GPR15L as the chemoattractant. So this is the data similar to what you saw before. We've got good migration that disappears when you remove polysia. If you add soluble polysia, we see a big decrease in the amount of migration, and then a little bit extra decrease when you also treat with the endosialidase. If you add even more polysia, that decrease is even more significant. So it's a dose-dependent decrease in migration in the presence of soluble polysia. We can also inhibit migration using an antibody towards GPR15L. So just comparing this to this, we see a decrease in the migration in the presence of anti-GPR15L and a more substantial decrease in with a higher concentration of anti-GPR15L. So this data told us that the migration of these T cells truly is dependent on polysia, and that GPR15L is likely binding to T cells. And we have a lot more data to show that interaction directly. But what I want to show you now is that we wanted to determine whether or not this was actually relevant in vivo in some sort of pathogenic inflammatory condition. And so what we looked at was psoriasis in collaboration with, again, Mo Osman and Robert Niedecki here at the University of Alberta. So what we're looking at is spatial transcriptomics for a skin section of a psoriasis lesion in patients before treatment and then after receiving, I think it was anti-IL-17 treatment. So this is after the lesion is resolved. We see lots of GPR15L in the skin. This was known, it was in the literature, that GPR15L, the ligand expression, is much higher in psoriatic lesions and is substantially decreased after resolution of the inflammation. We also see GPR15, so you're looking for the orange and red circles. These are circles where there's GPR15 transcripts present, and again, they virtually disappear after the resolution of inflammation. And then SD8-SIA4, there is a lot of SD8-SIA4 mRNA present, and it's overlapping. I haven't shown the data here, but it's overlapping with an immune cell compartment and overlapping with most of the GPR15 spots. And again, after the resolution of inflammation, there's virtually no SD8-SIA4 present. So we're really excited about the connection between polysialation, cell migration, and cell migration, particularly in T cells. So coming back to this is what we originally started off on, and unfortunately, GPR15 has nothing to do with systemic sclerosis. And so what we want to do now is to do the exact same thing, but with T cells from patients with systemic sclerosis, to understand how these T cells are migrating to the skin. And not just T cells, we're interested in other cell types as well. But since this didn't work, I want to show you a very quick secondary story that actually follows on from the first methodology we developed to identify polysialylated proteins, because this is where we are making some really cool headway in scleroderma. So if you quantify the amount of polysialic acid in scleroderma, we see that there is, you compare it to healthy controls. In patients with scleroderma, the amount of polysialic acid is elevated. And in females or women, the levels go up in the limited scleroderma, and then in the more severe form, they go up even higher. So there is a very strong correlation between the amount of polyscia and the amount of inflammation in the tissue, in the actual tissue. So the modified Rodnan skin score, which is a measure of disease activity in the tissue in scleroderma. There's a very, very strong correlation in women, not in men. So this is the polyscia in healthy controls, age-matched controls. It definitely goes up a little bit in men with scleroderma, but there's no correlation with disease severity or with the skin score. And so we really wanted to understand this better, thinking that maybe if we could figure out what these proteins were, we maybe would have a biomarker for who might progress to having a more severe form of the disease. And for reasons that I'm not going to go into, that would be really important in scleroderma. So if we use that original methodology that I showed you to identify polysialylated proteins, in serum, in healthy serum, we found CD56, NCAM, and SD8-SIA4. It worked really well for these two proteins. But in scleroderma, we still see these two proteins, but now instead of seeing this nice volcano plot, symmetrical volcano plot, we're starting to see weirdness here. There are some proteins that are clearly they're much more present in some samples compared to others, but they're not consistent. And we didn't know if this was because we didn't have a lot of materials. Scleroderma is a rare disease, and so getting a lot of material to work with can be a challenge, or what was happening. What we knew, though, is that the methodology that we were using had a complication to it. And that complication was the fact that when we do the proteomics analysis, we're sending the streptavidin-coated beads with endo-N with whatever polysialylated protein is attached. And so for every endo-N that's binding a polysialylated protein, we also see peptides from endo-N. And so if you know anything about proteomics, you know that that would actually suppress the signal from other more rare samples, proteins that are in the sample. And so we devised a new method to isolate polysialylated proteins that still uses streptavidin beads, but no longer requires that endo-N lectin. And the way that we do that is to take advantage of the fact that the bacterial polysialyl will transfer modified CMP sialic acid to any 2,8-linked sialic acid, but it will not touch a 2,3 or a 2,6. And so we showed that we can transfer biotinylated CMP sialic acid to proteins. And I won't spend too much time on this, but we can isolate these proteins on streptavidin and agarose, and then we can do proteomics. And so this is the data that we got from healthy serum. We still see NCAM, which is fantastic, but now we're seeing a lot more proteins. And one of the advantages of this assay is actually that it's not just polyscia that we'll see, it's also the short oligoscia. So much less is known about oligoscia because we just don't have the tools to enrich it, to isolate it, any of that. And so this is one of the first methods that will also allow us to detect short oligoscia chains. And you can see there's a ton of proteins here. We're really excited to start working our way through them and figuring out what's interesting to us, what's interesting to other people, that kind of thing. So when we did this with scleroderma, we also got some fantastic data. And when we compare scleroderma to healthy controls, so now we're looking for proteins that are specifically dysregulated in scleroderma, there are two proteins here. And unfortunately, I can't tell you what these two proteins are. We're still working on them. But what I can tell you is that this methodology is really robust. It gives us just fantastic data, and we're really excited to follow up on what's happening here. So with that, I want to thank the people who did the work. I have an incredible team. I'm really lucky to work with them. Thank the organizations who have funded the work, including GlycoNet. There's some GlycoNet swag here. I don't know if you can see that. Hopefully you'll all come to the Glyco 27 meeting that is in Edmonton in May and have the opportunity to see some more excellent glycoscience research. And I want to thank you very much for your attention. I'm happy to take any questions.
Yeah, thanks so much, Lisa. We have about 17 minutes for questions, so you can either ask them out loud or drop them in the chat, and we will get to your questions.
Questions? Makes my job easier. We can go have lunch.
Lisa, I can ask you a question. Andy Mason.
Hi, Andy.
How are you?
Good. How are you?
Good, good. I enjoyed that. So some of those proteins that you were pulling up look like an ER stress response.
Sure do, don't they?
EIF, and you've got a lot of ribosomal proteins, etc. So how would you sort of integrate that into your hypothesis of what the salo proteins are doing?
Yeah, that's a really good question. I don't want to spill beans too early, because Mo now has quite a bit of data. Charmaine and Lamia have been doing some really cool experiments that show that there is a connection between polysia and the unfolded protein response and metabolic dysregulation. And so stay tuned. There's definitely a connection there.
Okay,
Well
so that's pretty much why I was asking the question, because we see the same issue with primary biliary cholangitis that shares many of the features with systemic sclerosis, except our pathology is in epithelial cells, and then Mo's pathology is more vascular and fibrosis related.
Mm-hmm. Yep. We've not, I mean, it would be, so actually, I'm not aware if anyone has looked. So there is a lot of cancer data that shows that doesn't matter where in the body you are, polysia shows up in cancers. I don't know if anyone has looked to see if there is a propensity for polysia to show up in particular cell types that become transformed, or if it is a general feature of just that transformation. So there's two things that are happening. One is the dysregulation of the immune system, and the other is the dysregulation of the transformed epithelial cells or the fibroblasts themselves. And certainly in scleroderma, both are important. I don't know if that's the case in other inflammatory diseases, but it wouldn't surprise me if that were the case. If you've got some epithelial cells, we could look at them.
Well, you see, that essentially that's why I was asking, because the commonality and the reason we got working with Mo is that the two autoimmune diseases share some very, very specific biomarkers, such as anti-mitochondrial antibodies, changes in metabolism, et cetera, So we've looked for the virus that we've linked with primary biliary cholangitis in patients with systemic sclerosis and found it's there. So then the question is the virus linked with some of the phenomena you're looking at? Because it is an oncovirus that changes intracellular metabolism into more of a cancer metabolism, et cetera, et cetera. So a lot of the features you're pulling up, I sort of prick my ears up when I hear you talking about breast cancer cells, because it is essentially a breast cancer virus. So
Yeah, okay.
that was where my question was coming from, but
Yeah.
obviously we could go on and on down this line, but we could talk offline later if you like.
That's good.
Thank you.
Thank you.
Any other questions? If not,
I have
oh,
one question. Thank you very much for that great presentation. I noticed that you mentioned in the beginning the kind of a paradoxical relationship between the amount of polycia that you have on T cells as they're getting more activated. And you touched on the effect of the PSA on the migration part of T cells. But I was wondering if you also have looked into how the polysialic acid is actually taking part in the immunomodulation part on T cells by like engaging with like Siglex either on different cells or on the same cells, or maybe even like if polysialic acid effect is affecting something inside the cells, or is it just like merely just a physical interaction with the charges and everything that's causing the migration to change?
That's a great question. So there are no known Siglex on a T cell, known. And so the obvious way that you might go about, knocking out a Siglec and finding out if polycia is inhibitory in the way that it's inhibitory in a macrophage, is not possible for T cells. There is some data in the literature that suggests that loss of polycia increases T cell activation. That is not the case in our hands. You can remove polycia and there's no differences in proliferation, in activation, at least in vitro, with synthetic activators. Maybe in vitro, in a more biological context, that might be true, and that's something we're actively looking into. As far as the how does it actually affect migration? It's a really good question. Does polycia do more than just act as a landing pad for GPR15L? Does GPR15 itself need to be polysilated, or is just having polycia present on the cell surface sufficient to sequester GPR15L essentially, or bring it in close contact with GPR15? We don't know the answer to those questions. We're working on that for sure. Yeah, I think that's all I can really say, is that these are questions that we're also asking ourselves, and some of them we're actively working on, but I don't have any answers for you just yet.
Thank you. So basically just the increasing amount of PSA as the T cells are getting activated, because I kind of assumed that when I saw the data that it's somehow maybe in addition to the migratory effect is trying to somehow compensate, like maybe like oppose the like overstimulation or overactivation of T cells in some way. And I just want to see if there's any data on that or not.
Yeah, so one of the things, and this is kind of why I mentioned timing right at the beginning, is we published the immune cell data. It's not published, it's in bioRxiv, where we working on getting it published. And we took naive CD4 positive T cells and we activated them in vitro. And you get a little tiny bit of polycia after 24 hours, but it's more the 48 to 72 hours that you get polycia, really robust expression of polycia. And so if you're thinking about cell migration, and what actually happens in a body, that's a very delayed response. If a T cell has seen an antigen, the proliferation starts to happen pretty quickly. Pretty quickly. You don't actually start to get massive proliferation until later. That's in a naive T cell. I don't know about in a antigen experienced T cell how long it takes that activation to happen. But what we're thinking is that polysialation is important for directing the T cell where to go in the body. So if you polysialate the GPR15L, it will then go to polysialate GPR15L, it will then go to like the mucosa or the skin, depending on where you are or where the T cell is. Yeah, so it could be just a timing thing. It could be that it gives it some specificity for where it's going to go. It could be that you get that activate, like the signals that are needed to get T cell activation happening on a naive T cell at least wouldn't encounter polysia, but then 3 days later you have polysia. So is that then preventing that cell from reactivation or increasing the threshold for reactivation? That's a possibility, and that's why we see some on memory cells. If you then reactivate the cells, do they immediately lose polysia? The effector cells certainly have slightly less polysia. So I think timing is going to be a really important parameter in understanding what polysia is actually doing in a biological context. I'm not sure if that answered your question. I felt like I rambled.
No, that was great. Thanks so much.
Any other questions? If not, Lisa will be at the GlycoNet Symposium in May, so sign up if you haven't already. It's our little plug of the day, and you can ask her more questions there. But if no one else has any other questions, we'll wrap it up. And thank you so much for joining for one of our first GlycoNet webinars of the summer season. So have a great weekend, everybody. Thank you for joining.